All right. Hello, everyone. My name is Yezen, and I'll be summarizing this paper. It's basically a phase one randomized placebo-controlled study on low LSD doses in healthy older volunteers. Here's our outline. I'd like you to keep these questions in mind throughout the sections. So we'll start out looking at the relevant facts, and then we'll bring those facts together to understand why LSD effects in the elderly were studied here. We'll look at how these effects were quantified, and we'll look at the results and what the researchers concluded on that basis. And then we'll consider some future directions in the area. Now, what are the relevant facts? So first of all, LSD is a serotonin 2A agonist. It also acts on pretty much all aminergic GPCRs. But most importantly, LSD and serotonin 2A agonists in general can increase the expression of BDNF in the brain. And this seems to happen whether doses are chronically administered or acutely administered. Furthermore, BDNF, most of you probably already know that BDNF has anti-inflammatory effects. It promotes neuronal survival, it strengthens synapses. It's generally a thing that's good for the brain in most cases. And one such example is this study right here that demonstrated that BDNF was capable of alleviating memory deficits in an Alzheimer's disease mouse model. So that's one chunk of information that suggests indirectly that LSD might help with Alzheimer's. And then a more surprising result, at least this surprised me, LSD apparently reduces pathological cleavage of amyloid precursor protein. But this was only shown in vitro with 3T3 cells, and that's a mouse-derived cell line. And yeah, so far we have 2 bits of information suggesting that LSD might help with Alzheimer's disease. And then as far as the memory and cognitive functions, which are usually impaired in Alzheimer's disease, serotonin 2A is heavily involved in both. So for instance, there's some literature suggesting that serotonin 2A activation can help fear extinction in animal models. And this, I think, is probably related to anxiety as well. And there's plenty of literature showing how serotonin 2A agonism can alleviate depressive symptoms and reduce anxiety. Now, how do all these facts come together to justify investigating LSD effects in the elderly? First of all, Alzheimer's disease mainly affects old people, and it's often comorbid with depression, and people with Alzheimer's disease often exhibit low concentrations of serotonin 2A receptors. Furthermore, the Alzheimer's disease phenotype is often characterized by a nasty positive feedback loop between amyloid beta aggregation and neuroinflammation. So given what we said so far about LSD and how it affects BDNF expression, and that 1996 result on serotonin 2A mediated reductions in amyloidogenic cleavage of APP, it's reasonable to assume that LSD could probably disrupt this feedback loop, right? So here's a simple schematic showing these feedback loops. And so far we have pretty good evidence to suggest that this part of the loop would be disrupted by LSD. This would be via BDNF, but I would imagine there are other mechanisms as well. And here we, from the facts I mentioned so far, it's only the 1996 result, but I mean, it's probably, it probably hasn't been replicated because scientific journals seem to discourage replication studies, unfortunately. But, you know, it's a possible thing, right? And the same amplification of BDNF that justifies this limb of LSD's actions in the brain would justify this one, ultimately reducing neuroinflammation. Now, neuroinflammation ultimately causes cell death, right? Via excitotoxicity and many other mechanisms, oxidative stress. So this would include serotonergic neurons. And speaking of oxidative stress tends to disrupt normal expression patterns. So even a serotonergic neuron that hasn't been killed off by neuroinflammation may exhibit reductions in serotonin 2A. And that is indeed what we observe in Alzheimer's disease patients. So I just showed you guys three basic plausible mechanisms that might alleviate Alzheimer's disease symptoms. Now, that's why we would look at LSD effects in older adults. Now let's look at the methods that they used in this study. So as I said earlier, this is a phase 1 randomized placebo-controlled study. The participants were 55 to 75-year-old healthy adults. And one of the key inclusion criteria here was that they had no experience with LSD within the past 5 years. This was to omit any possible confounding effects on the results. And then this sample consisted of 48 individuals, and they were divided into 4 cohorts randomly. And the cohorts were each given a particular dose 6 times. Each pair of doses was separated by 4 days, right? So at 4 days intervals, the participants were given a dose. Here are the doses: 5 micrograms, 10, 20 micrograms of LSD, and of course the placebo group. And all of that was administered orally in a solution. The measurements they took were, they did some standard measurements like ECG, blood pressure, things like pulse rate, standard stuff you would see in phase 1 clinical trials. And they also included balance and proprioceptive assays, as well as cognitive assays. These ones focused on visuospatial memory and other visual tasks that tested cognition in a variety of ways. And the participants were also given questionnaires to assess their subjective states. So it's definitely worth mentioning at this point that all these doses are subperceptual in most cases. So uh, there was no concern of the participants having any intense subjective experiences. But nonetheless, these questionnaires were very informative, as we'll see later on. And dose response, how these measures were affected by dose, um, those correlations were established by classic pharmacokinetic measurements, like plasma LSD concentration over time, area under curve. This is uh, this is informative in that it shows us how bioavailable LSD is, how much absorption happened, and things like that. Peak concentration time, how fast LSD is absorbed into the bloodstream, and the assays listed above, of course, balance, proprioception, cognition. Those were all used together in order to produce uh, correlations between LSD dose and performance on these assays, and of course, answers on the questionnaires. And so the first result, uh, first set of results was on safety and tolerability. Most notably, headaches were observed. Mild to moderate headaches were observed in the treatment groups more so than in placebo groups. Um, Interestingly, the middle dose cohort, 10 micrograms, uh, exhibited the highest incidence of headaches. It was so remember I said the sample consisted of 48 individuals. Each cohort was 12 individuals. And this cohort, 6 of those individuals uh, reported headaches. So that's 50%. And that's interesting when you compare it to the lower cohort, the 5 microgram cohort, and the high dose cohort, the 20 microgram dose cohort. This is the highest number of headaches. So I thought that was worth mentioning. That's pretty interesting, I think. In the highest dose cohort, the 20 microgram cohort, uh, the incidence of headaches was just around 25%. So I wonder what's going on here. But luckily, no one discontinued the study due to adverse events. And this is pretty much the only serious concern revealed by the safety and tolerability data. It's just headaches. Pretty much everything else, there was no significant difference between placebo and treatment groups. But the questionnaire data did reveal one interesting detail as well, uh, which is alertness. So the participants who were given LSD doses did report feeling less alert, and these reports were linearly associated with dose. So there is definitely a dose relationship here. But as you may have noticed, I did write self-perceived, and that'll be, the reason for that will be apparent in a moment. So dose response, the results revealed, the pharmacokinetic measurements revealed that repetitive dosing was effective, right? So there was no reduction in the capacity of the participants to absorb LSD the more LSD was given. I'm not sure if I mentioned this earlier, but they were given 6 doses separated by 4 days. So this kind of regimen, theoretically, it should work based on this pharmacokinetic measurements, and you wouldn't need to increase dose to deal with absorption concerns or anything. So that's good. And there was no plasmatic LSD detected with the 5 microgram group. Uh, it's not much of a surprise. It's a very low dose. Probably very little of those 5 micrograms got through. It's probably very difficult to detect whatever did get through. Perhaps nothing got through. Who knows? Uh, but in any case, my personal opinion is that 5 micrograms is probably too low a dose to have any kind of effect on symptoms if this treatment actually does work on Alzheimer's disease. The pharmacodynamic side of things, they only used proxies in this paper, and those were the cognitive, balance, and proprioceptive assays. And interestingly, there were no differences between treatment groups and the placebo cohort. So here we have participants reporting that they feel less alert, but they're performing just as well as placebo participants in these cognitive, balance, and proprioceptive assays. So that's interesting. I would think that something that may have caused that, one possible cause of that effect, this result, is that these older adults are showing up with the impression that, oh wow, I'm participating in this study with LSD. LSD is this crazy illicit drug. So I would guess that this prior result here, the alertness result from questionnaires, that was probably influenced by participant expectation. But who knows? Anyways, the researchers made a few conclusions here. Firstly, low dose LSD is safe and tolerable in healthy older adults. There's a minor concern when it comes to headaches, and one possible mechanism that could account for this, it is a real, well-established mechanism, would be that serotonin 2B agonism is the culprit. So disruptions of heart rate and blood pressure generally have been shown with serotonin 2B agonism. And LSD does have some partial agonism to serotonin 2B, but it does mainly act on serotonin 2A. So here's one possible mechanism which may account for the increase in headaches. And then in terms of dose response, the pharmacokinetic data demonstrated quite clearly that repeated oral doses would be effective with low dose LSD. So that's not a concern for future clinicians if this actually makes it through all clinical trials, which I certainly hope it does. I hope it's helpful. And then the self-perceived reduction of vigilance that we saw in the questionnaire data, even though it doesn't impair cognitive, balance, or proprioceptive function, the authors did conclude, I think quite reasonably, that it's something worth investigating and considering because it might have an effect on lifestyle. It probably doesn't feel great to feel drowsy or dizzy, and feeling like your alertness is compromised probably doesn't have the best effect on your lifestyle. But if this treatment is demonstrated to have a robust therapeutic effect, the therapeutic effect, I would imagine, probably outweighs this concern. And again, I would guess that a big part of this self-perceived reduction in vigilance is probably just the expectation of the participants. Now, what comes next in this area? Obviously, the most important thing, the number one priority, is to demonstrate a robust therapeutic effect of low-dose LSD in people who are actually affected by Alzheimer's disease. And this is exactly what these authors plan to do in phase 2 clinical trials. And hopefully they get through that. But ideally, I would think that the best thing would be a long-term study to see how this effect, how this potential therapeutic effect plays out over time. We'd need a longitudinal study. We'd need a much larger sample. Remember, we only have 48 participants in this study. So really far from statistical power, right? So we need a much larger sample. We want a double-blinded and randomized study, of course, placebo-controlled. We don't want neither the researchers nor the participants to know who's getting what. We don't want any bias in assigning certain participants to certain dose cohorts. And we want to measure these therapeutic effects on symptomatologic scales. I also thought it would be interesting to see how this potential therapeutic effect would instantiate itself at different stages of the disease progression. After all, it might be the case that low-dose LSD treatment is only effective, say, in the initial or middle stages, but not late stages of Alzheimer's disease. It's probably worthwhile to investigate that. And furthermore, it's worth mentioning that all the evidence that suggests these mechanisms of action have been found in animal models or in vitro, in this case at least. The 1996 results showing reduced amyloidogenic APP cleavage, that was conducted on a 3T3 cell line, and that cell line is derived from mice. So we want to know if these mechanisms actually take place in human cells. So our options, the options I suggest at least, would be cell cultures and brain organoids, because we'd have much more freedom in manipulating things and really getting in there and seeing if these mechanisms are at work in a robust manner. It'll be quite difficult, I would think, to measure these effects in an unambiguous way in actual people. You have ethical concerns. So, you know, cell cultures could be useful. You could do a thing with induced pluripotent stem cells. You know, you differentiate some of them into neurons, some of them into microglia, and some into astrocytes. You try to reflect the natural ratios and distributions of these. So that could be useful, but most useful, I would think, is brain organoids. And a common problem with brain organoids is that the center of the organoid would be necrotic because there's no vasculature. But recent research has shown that you can get around this issue by taking that human-derived brain organoid and implanting it into a rodent's brain. And apparently, it becomes fairly integrated into the rodent's brain, and you have cross-innervation between the organoid and the host tissue, and you also have vascularization of the center of the organoid. So that should be I think that would be a very good model to mimic in vivo environments in the actual human nervous system. And that should help in verifying whether these mechanisms take place in human cells. And finally, I just want to thank Fomity et al. For their great contribution to science and human well-being. These people are doing pioneering research on psychedelics, and I think we should all be grateful to them. Especially the work on depression, treatment-resistant depression. I mean, it's very, very promising. And also, big thanks to Alex Gray. Alex Gray, a brilliant artist. You may have noticed on the overview slide, there was that beautiful painting. That was Alex Gray. So please check out his work and support him. And last but not least, of course, thank you to Dr. William Ju for giving us this opportunity to showcase our abilities. I think this video journal idea is absolutely brilliant. I've always wanted nice, direct explanations of scientific papers in video form, and I really appreciate that Dr. Ju is doing this. So thank you, Dr. Ju. And then here's a reference list. If you're curious about anything I said, you can take a screenshot here. And of course, here's my shameless plug. This is my website. If you're curious about anything I've said so far, if you want to collaborate on any projects you have in mind, feel free to visit my website and just scroll all the way down. My phone number is on there, my email is on there, and I have some of my prior assignments on there that reflect some of my interest. You can look into those if you're interested. And on that note, thank you all for listening, and take it easy.
Computational research on epilepsy tends to be focused on the modeling of systems that can be induced into seizure states and the mechanisms that lead to that behavior, most often focusing on EI-based models of excitatory and inhibitory neurons. Recently, research has looked further into the relation of inhibitory systems and their relation to epilepsy, specifically how inhibitory neuron bistability can explain increased interneuronal activity prior to seizure. The cause of seizures and epilepsy tends to be related to excitotoxicity, the overactivation of excitatory neurons, most often glutamatergic neurons, hence why most treatments of epilepsy tend to be a variety of anti-epileptic or excitatory drugs, where a drug enhances the inhibitory neurons to counteract the overactivation related to seizures and epilepsy. Surprisingly, these anti-epileptic drugs are only effective 30 to 40% of the time when treated with patients with epilepsy. Recent research has shown that prior to seizure onset, interneurons showcase hyperactivity, indicating that interneurons, specifically inhibitory interneurons, may have a role in seizure onset. This hyperactivity may be useful, as it was well related to the researchers' previous work regarding interneuron systems and their capacity to be put into a bistable synchronous state, which could be related to the hyperactivity observed.
It was a bit of a serendipitous occurrence that this project had a lot of overlap with my PhD work, which was primarily in understanding the synchronization properties of networks with interneurons and how the properties, the single cell properties of the interneurons were actually having network level effects in how these networks synchronize and generate rhythms. And one of the things that we found in my PhD work was that, so there's type 1, type 2 classification of neurons based upon their phase response curve. And type 1 neurons in a network tended to have this bistable feature where you could have a network that from randomized initial conditions would be completely asynchronous. If I perturbed the system by essentially trying to force the system to synchronize, then it would preserve that synchrony moving forward. So we discovered the sort of network level bistability between an asynchronous and a synchronous state. Now, when I came to Toronto and I came to Krembil, you know, Francis and Taufik had some this sort of preliminary project that had been sort of in the weeds for a while, and I sort of came onto that and essentially saw what the network was and saw a experiment that one of Taufik's students had done, where he quite literally gave a optogenetic stimulus to two inhibitory neurons, and saw that by giving this optogenetic stimulus, which was very similar to this perturbation I had modeled in my previous work, you got these synchronous seizure-like dynamics in hippocampal slices. And it was quite literally a eureka moment where he sort of showed me this figure, and I pulled up my paper and I said, wait, so when you optogenetically stimulate, it's pretty much like doing this, right? And he was like, yeah. It's like, okay, I can model that.
Following previous research regarding mechanisms of inhibitory interneurons, the modeling was completed in an in silico model using an Zickovich model, with connectivities of 4, 8, 12, and 16%, in a system with 500 neurons. The study compared activity and synchrony of a model, control, untreated neuron network, and a model of a 4AP treated network, which is used in vivo/vitro models for epilepsy research, as 4AP treatment can lead to hyperexcitability, increased wheelbase, and other useful traits that would be useful in capturing an in silico model. When modeling the system, each neuron were given a set of random starting conditions before a fixed perturbation was introduced. When the perturbation was introduced, the range of weights and currents that induced a state of synchrony was wider in that of the 4AP model than that of the control. The frequency was also observed to be higher, most observable in the figure on the right, section C. Applying an external current, especially a fixed current, seems artificial. To showcase that this is plausible in a biological system, a Ornstein-Uhlenbach process was generated, a peak of which, from the OU process, was then used as an input to the system, akin to that of the perturbation. When the OU spike was introduced to the system, it was able to induce the stable, synchronous state observed when a fixed perturbation, an idealized perturbation, was introduced, thus signifying that it is plausible, if the OU process is plausible to that of background synaptic input, that this could occur in a biological state. The usage of the OU process, as explained by one of the researchers, is as follows.
With that OU process was this idea that, and this came about from, relationship with the literature, reviewers, etc., was a very logical question to ask of our paper is, okay, you've given the system this very idealized perturbation, but that's probably not going to happen in the brain or anything close to that, right? Mm-hmm. And so what we tried to do is said, okay, here is this OU process. And again, we just sort of chose the OU process because it's a very sort of generally used thing in the cortex. We sort of used cortical parameters from those papers we cited. And, we ran it and basically said, okay, if we're thinking about this system in the biological setting, the OU process shows that there are these fluctuations, right? And then we said, okay, here's a fluctuation that happens out of this OU process that sort of looks like our perturbation, not exactly, not quite as idealized, but somewhat similar. What if we use this more biologically reasonable, naturally occurring perturbation in our model? And lo and behold, that perturbation still induced the changes in bistability that we saw in the idealized setting. Okay. So that was that OU process was sort of a step towards showing the biological feasibility of this, showing that this isn't necessarily a function of the abstract setting that we're in, of the idealized perturbation that we've used. Something that is approaching the biology, and again, not exactly the biology, because again, we didn't exactly instantiate the OU process. Even that, you could argue whether or not that's a accurate, encapsulation of the noise, etc. But this is something that is used, as well established, and does sort of point us in the direction of this being biologically feasible.
Other characterizations of the model affect its ability to be driven into a synchronous state. The connectivity of the neurons influences the range of synchronous states greatly, where at 8% there is no significant difference between that of the control and the 4AP models. 12 and 16% connectivity models shows that the 4AP system transitioned to synchrony in a wider array of currents and inhibitory synaptic weights. When increasing the overall external applied current as compared to that of a idealized perturbation, the transition between asynchronous and synchronous states, both the rate at which that is conducted and the slope, as observed in the figures on the right, there is no significant difference in the rate of change between that of the control and that of the 4AP treated network, signifying that this induction into a synchronous state may be unreasonable for that of epilepsy, or rather that the induction of a idealized perturbation or even a OU spike would be more accurate in modeling that, or more generalizable. The major finding of this research showcased that there is a reasonable mechanism by which GABAergic interneurons may be driven into synchrony, and that capacity into a driven state of synchrony is dependent on a bistable transition, a perturbation. However, a general increase in external applied currents has no significant difference between that of a 4AP model and control, meaning it may be less generalizable. Future studies may find it useful to expand on this model by incorporating excitatory neurons or other types of inhibitory interneurons based upon cortical samples and calculations provided by Markram et al. Further means of study may research the capacity of interneuronal hyperactivity to be determined by EEG or related systems, such that they can inference the activity of these interneurons to predict the onset of seizure.
There's a lot of cool new technology coming about, with neuromodulation, with optogenetics, where you can really precisely focus things. So, if I were to design, a dream study, I would love to be able to sort of take a piece of tissue that was exhibiting, a seizure-like event, and then, desynchronize the inhibitory network via some sort of randomized optogenetic stimulus or something like that, and see what happens. Or do that probably more likely do that in the uptick to seizure. Because something that we have to be really careful about talking about this particular paper is that this paper is all about seizure initiation. When you talk about seizure, it's sort of split up into three separate sort of subfields: seizure initiation, seizure propagation, seizure termination. And one of the things I learned delving into the seizure literature is you have to be really, really careful about which one you're talking about.
Hello everybody! Thanks for tuning in today as I'll be presenting the research paper, Benefits of Physical Activity for Depression and Fatigue in Multiple Sclerosis, a longitudinal analysis published in 2019 under the Journal of Pediatrics. The progress of my presentation was conducted under the supervision of PI Dr. Ye. Multiple sclerosis, otherwise known as MS, is a neurological autoimmune disorder. The disorder targets oligodendrocytes, affecting myelination of nerve fibers in the central nervous system. Children with pediatric onset of MS, otherwise known as POMS, where the disease manifests under the age of 18, has shown higher rates of displaying fatigue and depressive symptoms compared to otherwise healthy children. Previous literature have proposed that this is the case, as children with POMS have shown greater complexity compared to adults. Differences include that they are more commonly diagnosed with optic neuritis, isolated brainstem syndrome, and encephalopathy. Therefore, symptoms including fatigue, depression, cognitive problems, vision impairment, and so forth are heightened in these children. Up to when this research paper was published, there has been previous cross-sectional studies that have reported association between lower levels of physical activity with higher levels of POMS, fatigue, and depressive symptoms. On top of that, cohort studies show that there is an association between physical activity and improved cognitive processing, such as memory. However, this area of research lacked longitudinal studies. These type of studies can help establish a time course of how physical activity can affect these symptoms. Therefore, this specific longitudinal studies aim to provide more context towards if lifestyle-based intervention can lessen depressive and fatigue symptoms of POMS. Before I present you with the hypothesis of this paper, I would like to point out that monophasic acquired demyelination syndromes, otherwise known as mono-ADS, refers to patients who experience only a single clinical event due to demyelination. This then leads them to experience symptoms of fatigue and depression, maybe dizziness, yet they do not display subsequent inflammation or further relapse as compared to children with POMS. Knowing this, it leads us to the two proposed hypotheses of the study. The first proposed hypothesis is that youth with POMS would experience depressive and fatigue symptoms at a higher level than mono-ADS children. Also, it would increase over time while the latter would stay static. Additionally, It is also proposed that youth who report higher levels of physical activity categorized as MVPA at any given point in the study would experience lower levels of fatigue and depressive symptoms. It is also noted that the authors speculated that children with POMS would engage in less physical activity than those with mono-ADS, and this is probably due to the fact that there are relapse and they experience further inflammation in their brain as opposed to mono-ADS children. So that brings us to methods conducted in this longitudinal study. The study collected and analyzed data from youths who consecutively consulted the Pediatric Neuroinflammatory Disorder Clinic located at St. Kitts, Toronto, Ontario, Canada, between September of 2013 to March of 2017. Details of inclusions and exclusion of the participants were not mentioned. However, all the youths would be diagnosed with POMS or mono-ADS according to the 2010 McDonald criteria. In the study, the data were collected through the form of self- or parent-reported questionnaires during each and every clinical visit. Depressive symptoms were reported through the Center of Epidemiological Studies Depression Children Rating Scale, which is a mouthful, while the fatigue was measured through the Pediatric Quality of Life Multidimensional Fatigue Scales. Note that both are symptom-oriented scales that can be turned into descriptive analysis with numerical data. So then by the end of the study, inferential statistics were then used to analyze differences between the POMS and mono-ADS cohorts. Physical activity level was recorded through the frequent Golden Leisure Time Exercise Questionnaire. Now, these questionnaires were administered at every clinical visit, aiming to record your physical activity level during a period of 7 days. In physiology, this data can then be used to rank individuals on a scale ranging from no activity, which is sleep, all the way to intense exercise. This is important for the study as we're trying to look at youths who report moderate to vigorous physical activity and if this has an association with their effect of depressive and fatigue symptoms. Additionally, clinical data collected was stratified for other demographics and clinical characteristics. This includes sex, age of onset disease, physical disability or EDSS, and psychosocial scores, which include cognitive fatigue, sleep, and rest. From these data, trajectory of depression and fatigue were modeled into graphs, while also adjusted for MVPA. From the results proposed by the study, this table sums up major results which indicates that clinical depression were greater in children with POMS and increased over time compared to mono-ADS children. Further investigation of the patient's clinical demographic also showed that high depression were observed in POMS youth who reported onset of demyelination at an older age. If you look further down, you'll also notice that total fatigue score decreased over time for both cohorts. This coincides with the initial hypothesis that POMS children would experience greater side effects of depression and fatigue compared to mono-ADS children. Looking at this graph presented to us, encoded in red are children with MS, and encoded in blue are children with mono-ADS. From reading this graphical data, you can see that children who experience more MVPA over the course of the study also experience lower level of depressive symptoms. However, the statement only holds true for both male and female participants in children with POMS. Furthermore, if you compare the clinical results of male to female patients, there's a stronger association of decrease in depressive symptoms with each unit of increased MVPA only in girls with POMS. So just like the previous graphical data, here you can see that encoded in red are children with MS, and encoded in blue are children with mono-ADS. However, contradicting to the depressive symptom graphical data, in here you read it as the lower the response rate, the greater the fatigue score. As a result, you can conclude that children with POMS who engage in greater levels of MVPA also had lower fatigue scores over time, although this association appears to be weaker compared to depressive symptoms. Further look into the subdivisions of total fatigue showed that there were no significant association between cognitive function, sleep, or rest and the level of MVPA participation in either group. This translates into, even though total fatigue scores were decreasing over time for both cohorts, the participation a physical activity level did not affect cognitive function, sleep, or rest. If we pull up both graphical data for both symptoms, you can see another jarring interaction. This interaction is found between sex and increased symptoms of depression and fatigue in pre-OMS girls, where you can see that they report greater signs of both symptoms and there were greater regression over time. However, this pattern does not hold true for boys. To conclude this study, in regards to the two proposed hypotheses, it is that there is an observed association between higher levels of physical activity and lower levels of depression and fatigue over time in children with POMS. On top of which, children with POMS have reported higher levels of depression compared to children with mono-ADS. Now, this report is relevant to the healthcare industry as pediatric demyelination diseases are observed to be associated with decreased quality of life, academic decline, missed school, isolation from society, and increased risk of suicide. So if this association holds true, then it could provide more context to therapeutic, non-invasive methods to improve the quality of life of these children and prevent future suicidality through lifestyle interventions. The critical analysis I have regarding this paper, which were also pointed out by the authors themselves, are that there are small numbers of male participants in this longitudinal study. Although unfortunately I was unable to hold a virtual meeting with my PI, through email I was able to ask, was the small pool of male participants in the study due to less referral of male patients, or were there more female youths with POMS at the chosen clinic? To which she replied, this is related to the small number of boys with pediatric onset MS. Further research did show that there is in fact an association between sex and demyelination disorders, as ratio of women to men with MS could be as high as 4:1. This means that the data in this paper is not necessarily skewed. However, further investigation where there is a population of balanced male to female ratio will provide deeper understanding of this association. Additionally, another issue I propose is that the physical activity level was self-reported or parent-reported through the GLTE questionnaire. The issue with this questionnaire is that it does not necessarily contain the intensity and frequency of physical activity. Therefore, once it is translated into the MET chart ranking physical activity levels, this may not be an accurate representation of the population. Furthermore, as it is a questionnaire, you are subjecting yourself to report and recall biases. In continuation with the interview I had with my PI, I was able to ask, it was noted that MBPA does not improve quality of sleep or rest. Do you believe that more in-depth analysis of REM and non-REM sleep will show otherwise? To which she replied, we are currently doing studies evaluating the relationship between sleep and physical activity in youth. We will have answers to those soon. I do not know what the relationship between REM sleep and physical activity is in this population. So I did ask more regarding her study regarding sleep and physical activity, and we shall look forward to that when the paper is published. However, I do propose that future directions of this paper could potentially look into the analysis of MBPA in relation to POMS children and their sleep quality. As mentioned by my PI, the ambiguous relationship between REM sleep and physical activity has yet to be explored. Furthermore, to be explored in children with POMS. Previous research has shown that lack of REM sleep is correlated to slower cognitive performance, episodic memory, or behavioral changes. In general, lack of sleep has also shown to affect mood and behavior. As such, this will be an interesting study to look into, especially in children with POMS, as they have not been studied before. And that concludes my presentation reporting on the paper Benefits of Physical Activity for Depression and Fatigue in Multiple Sclerosis. I'd like to thank my PI, Dr. Ye, for her time and supervision throughout the process, as well as Dr. Zhu for his guidance. It was an honor to be enrolled in this neurological seminar.
Hi everyone, my name is Jano Fan. Today I'll discuss about a rho kinase inhibitor research that was conducted by Dr. McRaven here at U of T. The title of my presentation is ROCK Inhibitors, a potential therapeutic approach for Parkinson's disease through the upregulation of parkin-mediated mitophagy pathway. Next, I'll provide a short background information about Rho kinase or ROCK inhibitors. ROCK is a downstream target of the small HTP binding protein Rho. ROCK has been identified to play many different roles in cellular functions, such as cell shape, motility, secretion, proliferation, and gene expression. Many researches have been done to explore its clinical implications. ROCK inhibitor was determined to have a cardiovascular benefits, and it was also capable of promoting survival in human embryonic stem cells. Recently, much attention was focused on targeting ROCK in neurodegenerative diseases such as Parkinson's disease, Alzheimer's disease, and ALS. Parkinson's disease is a neurodegenerative disease that is often associated with motor dysfunction due to the loss of dopaminergic neurons or the formation of Lewy bodies. Currently, there was no cure for Parkinson's disease. Involuntary tremors, muscle rigidity, and slow movement are often observed in PD patients. Furthermore, normal mitophagy pathway was also observed in Parkinson's disease patients. The diagram here shows the pathway of Parkin-mediated mitophagy. In the presence of elevated mitochondrial stress, PINK1 will associate with mitochondrial membrane and allow the recruitment of Parkin. Together, Parkin and PINK1 will induce mitophagy. The remaining question in the field is to find therapeutics that could prevent the degradation of the dopaminergic neuron. PINK1 and Parkin are the most extensively studied molecules with regard to the Parkin-mediated mitophagy pathway. The rationale of this paper was to identify small molecules that could modulate the activity of Parkin-mediated mitophagy pathway and to determine their effects. Below are three highest-ranking molecules. It was hypothesized that ROCK inhibitors will target the Parkin-mediated mitophagy pathway and exert a neuroprotective effect. It was important to find a novel therapeutic approach that can later be translated into clinic. Authors use a high-throughput small molecule screen to identify the molecules that will upregulate the Parkin-mediated mitophagy pathway. CCCP was applied to induce mitophagy pathway. In order to identify the localization of mitochondrial Parkin and cytosolic Parkin, HEK293 cell line was used to express GFP-labeled Parkin. MitoQC assay was also conducted to measure the damaged mitochondria in lysosome. mCherry and GFP are also fused to an outer membrane protein, OMP25, to observe the fluorescence expression. If red fluorescence was observed, it illustrates that damaged mitochondria are in the lysosomal compartment. shRNA is also generated against ROCK2. Its effects were later compared with SR3677, a ROCK2 inhibitor. The ROCK2 expression was validated by Western blotting. A different cell line, SHSY5Y cells, were also used to administer DMSO or SR3677 treatments, and to determine the involvement of HK2. HeLa cells were applied to examine the phosphorylation status of HK2 after DMSO or SR3677 treatments. Crystal violet staining and ATP levels were also used as indicators for cell viability. Crystal violet staining will stain for the live cells. In Parkinson's disease Drosophila model, paraquat, a neurotoxin, was applied to flies to observe Parkinson's disease-associated phenotype after SR3677 treatment. Mitosox is a chemical probe used to detect mitochondrial superoxide species. Fluorescence can be observed when the probe interacts with the superoxide species. TH-gal4-derived UAS park RNAi flies were generated to specifically knock down parking in dopaminergic neuron and evaluate the climbing activity after co-administration of paraquat and SR3677, a fly model with specific mutations that causes the increased level of reactive oxygen species was also generated to test the effect of SR3677. The results showed 2 highest-ranking ROCK inhibitors and 1 ROCK2 inhibitor, SR3677. SR3677 is a ROCK2 inhibitor that selectively targets the brain tissue. The recruitment of HK2 was also determined. HK2 is a positive regulator of parking. On the bottom right graph, we can see the HK2 was mainly localized on the mitochondrial membrane instead of the cytosol after SR3677 treatment. Enhanced lysosomal degradation can also be observed on the bottom left diagram. After administration of SR3677, more red fluorescence can be observed, which indicated that there were more mitochondria associated with lysosome. A neuroprotective effect of SR3677 was also observed in parking and ROCK-dependent manner. ROCK is a Drosophila homologue of ROCK, which share the greatest sequence similarity. In the graph B, we can see cell viability was increased after SR3677 treatment in a dosage-dependent manner. We can see increased cell viability was correlated with SR3677 concentrations. More viability can be seen when more SR3677 was added. In parking knockdown flies, co-treatment of SR3677 with paraquat showed the improvement of climbing activity. Tyrosine hydroxylase GAL4 derived UAS ROCK RNAi flies showed no improvement of climbing activity. This indicated that the neuroprotective effect is ROCK dependent. This study answered the previous question. Small molecule ROCK inhibitors were identified. The ROCK inhibitor-dependent neuroprotective effect was also upregulated in parking medium mitophagy pathway. Hence, ROCK can be targeted and may serve as a therapeutic approach. Extensive researches could be done on other ROCK inhibitors. Also, the study can be translated into clinic, which definitely will bring hope to Parkinson's disease community. In the future, replication of this study was important before translate the study into clinic. Also, other ROCK2 inhibitor can be identified and examined. HK2 inhibitor can also be tested to see its effect on the parking medium mitophagy pathway. Lastly, the neuroprotective effect can be examined in different neurodegenerative diseases. Relevant studies also report the neuroprotective effect was seen in Parkinson's disease and ALS. In those diseases, ROCK is often increased in microglia cells, which lead to the inflammatory response. ROCK inhibitor could reverse inflammatory response and create a neuroprotective environment. ROCK inhibitor Vasudo was also exists in the clinic for treating pulmonary artery pressure in Japan. Furthermore, Vasudo was also determined to have a high safety profile. Hence, in the future, it could be directly tested in the Parkinson's disease model. Thank you for listening to my presentation.
Hello, my name is Becky Gross, and today I will be presenting the following article for the Journal Club. A comparison of regional brain volume and white matter connectivity in subjects with stimulant-induced psychosis versus schizophrenia by Peter Alexander, Christina Gikas, and colleagues, published in 2019 in Psychopharmacology. All right. So in this presentation, I will go over the background, methodology, results, discussion, and finally future directions and Q&A with one of the principal investigators of this article, Christina Gikas. Okay, so first, let's just start with some background information. Broadly, the research we will discuss today will focus on comparing symptom severity between three groups, the groups being stimulant-induced psychosis, schizophrenia with comorbid substance dependence, and schizophrenia without substance dependence. And before delving into the literature, we'll just define some key terms. So all three groups fit under the umbrella diagnosis of psychosis. So I'll just examine what psychosis is first. So people with psychosis have trouble or are unable to perceive reality, and as a result, they experience hallucinations, delusions, and disorganized thoughts. Psychosis itself can manifest due to many different reasons, and it's considered a condition rather than a disease by itself. And disorders that include psychosis as one of their symptoms are generally grouped in the DSM and ICD as psychotic disorders, which inside it includes both schizophrenia and substance-induced psychosis, among others. So schizophrenia is a neurodevelopmental disorder, typically manifesting in early adulthood, that is, and it's marked by genetic vulnerabilities coupled with environmental insults. Meanwhile, substance-induced psychosis symptoms manifest as a direct result of drug use. So in terms of substance-induced psychosis, not all substance users experience substance-induced psychosis. And for that reason, it is thought to be a result of genetic vulnerabilities as well. The last important point about substance-induced psychosis is that psychotic symptoms can be induced by many different substances. So, for example, a lot of the recent research is really focusing on marijuana-induced psychosis, but today we will actually focus specifically on stimulants. So now that we have all the main things introduced, along with the basic definitions, we can compare and contrast the two forms of psychosis straight on. So in terms of similarities, both schizophrenia and substance-induced psychosis are characterized by positive and negative symptoms. So positive symptoms are symptoms of psychosis, like we discussed, hallucinations and delusions, while negative symptoms are a deterioration of normal functioning. So that is something like blunted affect, social withdrawal, apathy, and etc. Other similarities include genetic vulnerabilities, also similar marking of gray matter structure abnormalities and white matter track differences. And the last similarity we'll discuss is recent research highlighting similarities in birth complications, psychomotor functions in early childhood that is more similar between substance-induced psychosis and schizophrenia than substance-induced psychosis and other disorders like substance abuse in general. In terms of the differences, one of the main differentiators is etiology. So the etiology of schizophrenia is very poorly understood, and so it's understood to be a lifelong neurodevelopmental disorder. Meanwhile, the etiology of substance-induced psychosis is a bit better understood. It's very tightly connected to substance consumption, so it's a bit more clear. A second main difference is the time frame. So in terms of substance-induced psychosis, many of the symptoms are transient and will disappear after the cessation of the drugs. Alternatively, sometimes the symptoms are actually a result of drug withdrawal and will cease with the resumption of the drug. So the time frame given for the resolution of substance-induced psychosis is usually somewhere between 1 to 6 months. Schizophrenia, on the other hand, is marked by being a long-lasting chronic condition, and it's actually not even diagnosed until the 6 months mark. So you cannot be diagnosed with schizophrenia unless your symptoms are at least 6 months into it. And it's due to this time component as the main differentiator that there is some kind of like intense debate of whether substance-induced psychosis and schizophrenia are very distinct conditions. Maybe they share some kind of neural pathway networks, or simply substance-induced psychosis may be a primary psychosis condition that is triggered by drugs. So this kind of speculation is especially relevant since many patients with an initial substance-induced psychosis get diagnosed with schizophrenia later on as symptoms persist, particularly if drug consumption remained consistent after the initial psychotic episode. So in the research today, the authors were motivated to further explore the differences between the two disorders in order to determine if there is a shared neural pathway or whether they are distinct. They've noted that there has only been one article to date comparing the two groups using neuroimaging techniques, and actually none where participants had a shared environment. So the author really aimed to rectify that in their current research, comparing the two forms of psychosis head to head. And of particular interest, the researchers focused on comparing the difference in symptom severity between the two groups. In terms of the hypothesis, the authors hypothesized that on a surface level, in terms of psychotic symptoms and neurocognition, the two groups would appear similar. But individuals with schizophrenia would actually have a more neuroanatomical deterioration. And they hypothesized that might be because of the neurodevelopmental origin of schizophrenia. Okay, so the researchers divided the 96 participants into 3 groups. 39 participants were in the stimulant-induced psychosis, 39 in schizophrenia with substance dependence, and 18 in schizophrenia without substance dependence. The researchers included the schizophrenia-dependent group as a form of control to control for the effects of substances on the brain. All participants were part of a separate longitudinal study and were all living in a single-room hotel in a poor neighborhood in downtown Vancouver. The participants underwent a battery of tests, starting with general physical health and blood tests, to scales assessing their negative and positive symptoms, to neurocognitive testing, assessing their verbal learning, memory, inhibition, mental flexibility, and affective decision-making. And lastly, they also underwent some neuroimaging, particularly MRI and DTI. Okay, so in terms of the results, there were significant differences in terms of the gender composition of each group, with the stimulant-induced psychosis having the highest number of women, followed by the schizophrenia non-dependent, and last by the schizophrenia dependent. There are also differences in antipsychotic intake, with both schizophrenia groups using more antipsychotic medication compared to the stimulant-induced psychosis group. And lastly, there were differences in substance use, with heroin use being significantly higher in the stimulant-induced psychosis group, and stimulant use significantly higher in both the stimulant-induced psychosis and schizophrenia-dependent group. Other than that, health and education were comparable between all 3 groups. Next, in terms of the psychosis symptom severity, there were significant difference in the positive and negative symptoms, with schizophrenia dependent group having the most positive symptoms, followed by schizophrenia non-dependent, and last by the stimulant-induced psychosis group. In terms of negative symptoms, the schizophrenia dependent group also had this significantly more negative symptoms than the other 2 groups. And there weren't much difference between the schizophrenia non-dependent and the stimulant-induced psychosis. With regard to neurocognition, none of the neurocognitive tests actually achieved significant differences between the groups, but there was a modest, non-significant trend of worse cognition performance for the schizophrenia-dependent group compared to the other 2. And lastly, in terms of neuroimaging, the MRI scans revealed larger volume of left parietal temporal lobe in schizophrenia non-dependent group compared to the dependent group. And with DTI, the DTI scan revealed lower FA and higher RD levels in many different brain regions in the stimulant-induced psychosis group compared to the schizophrenia-dependent group. And an exploratory analysis controlling for both heroin use and antipsychotic medication use was conducted in order to see if the results still remain significant. So in terms of the heroin use, when heroin use was controlled, the results still remain significant, and the FA and RD rating between the groups still differed greatly. However, when the antipsychotic medication was controlled for, the significance disappeared. Okay. So overall, the research elucidates a number of factors related to symptom severity. First, it appears that individuals with schizophrenia and comorbid substance dependence have more positive and negative symptoms, as well as possibly worse neurocognition functioning compared to individuals with stimulant-induced psychosis and non-dependent schizophrenia. In addition, the substance-dependent schizophrenia group also had less left parietal temporal lobe volume, which is interesting, I found, because these areas are usually associated with speech and verbal memory, and neurocognitive testing measuring those abilities did not actually differ in the groups. So some of the hypothesized reason as to why there is such a difference between the schizophrenia dependent group and the rest may be due to the additive effect of heavy substance use and schizophrenia. Another possibility may be that individuals with substance-dependent schizophrenia may be more mentally ill as a group in general. In terms of the neuroimaging results, the researchers predicted greater neuroanatomical vulnerabilities in brain scans in the schizophrenia group compared to the stimulant-induced psychosis group. And interestingly enough, we saw the exact opposite occurring in the DTI imaging. So the results revealed lower FA and higher RD on the anisotropy. And lower FA generally correlates with poorer cognitive performances, while high RD is associated with demyelination. And these results become insignificant, as we discussed, once antipsychotic use is controlled for. So based on this, we can see some really promising evidence that antipsychotic medication may be useful in preventing neurodegeneration, and that all individuals suffering from psychotic disorders may benefit from using them. So next, we can discuss some of the strengths and limitations of this research. So overall, I think this research is incredibly well done. We previously in the background mentioned that this study is actually one of the very few that directly examines the differences between stimulant-induced psychosis and schizophrenia. And moreover, it is actually one of the even fewer that controlled the effects of drugs on the brain by including a substance-dependent schizophrenia group. Another important aspect the study controls for is environmental factors. So by ensuring that all the participants live in similar conditions and have similar backgrounds, the researchers avoid confounds that could have influenced the results otherwise. And the last strength we'll discuss is that this research had an impressive range of comprehensive tests measuring symptom severity in several different ways. And each of these kind of tests help us clarify the degree of differences between the groups. In terms of limitation, there were a few. So for one, there was no control group that is free of psychotic symptoms. So this can be either a control group that is substance dependent but psychosis free, or you can include a control group that is substance free and psychosis free, or you can even use both. By using these groups, we could really see how each disorder differs from the normal population and to what extent each kind of like group is compromised. The next limitation I will discuss is in the background, we mentioned that kind of like the symptoms of psychosis in individual with substance-induced psychosis are really heavily associated with the recency of their drug use. So some of the participants in this research didn't consume the drugs within the past week, so there might be some kind of compromise in the data. Okay, so in this next section, we'll have a Q&A with Dr. Gikas. So I managed to get in touch with Dr. Gikas, who is one of the principal investigators of this research, and she was kind enough to answer my questions via email. And in this section, I will just share with you some of the questions that I had and her responses. So my first question was this one. From my reading on substance-induced psychosis, I found that there are many different types of substances that could lead to a psychosis. What made you interested in looking into methamphetamine or stimulant-induced psychosis in particular? And her response was, This class of like stimulants seem to be among the more common substances related to substance-induced psychosis, and stimulants are also commonly used in marginalized cohort, which is the cohort that has been studied here. The next question I had to do more with environmental factors. So I found it really valuable that the environmental factors were matched between the three groups while reading the study. And in the paper, it was mentioned that the study population came from a lower SES background in Vancouver. But recently there has been actually a lot of talk about the increase of substance abuse in higher SES areas due to the opioid crisis. And eventually a lot of these people who were affected by it transitioned to stimulant groups like cocaine. Is it possible that there is symptom severity differences between SES groups? Would SES be a factor here? Etc. So the response to that was that was a very difficult question to answer, which is fair enough. There are a lot of things that are confounded with SES, which make it difficult to study on its own. And symptoms themselves are not necessarily directly tied to SES, but rather to more direct factors like substance use pattern, medication, type of psychotic disorders, and other individual differences. The last question I had was related to gender. So did you do any exploratory analysis on differences in symptoms based on gender? Do you expect any gender differences? And the response to that was, I believe that there are biological sex differences in the distribution between the psychotic group in this particular sample, though they do not measure gender here. The reason they do not measure gender here is because there was an adequate amount of power to examine this further. And in addition, I am not familiar with any literature on sex differences on type of symptoms presentation. So this kind of nicely brings us to the conclusion and future direction. So based on the kind of question I asked, you can probably see that I am very interested in exploring how different subgroups may be affected by the disorder differently. So for example, we can bring out the gender question. There is some evidence that I have recently saw in a review by Vokey and colleagues suggesting that females with psychosis exhibit more affective symptoms and less negative symptoms. So I would really be interested in the future to explore how gender would manifest differently in terms of like neurocognitive tests and neuroimaging tests, and if any of these would be apparent to us. Another really interesting future direction we can explore would be conducting a longitudinal study on individuals with substance-induced psychosis and trying to identify possible markers predicting which individuals would later on develop schizophrenia and which one will not. So some interesting research that has been done regarding this examined the likelihood of individuals with substance-induced psychosis to develop schizophrenia based on the primary drug they abuse. So really interesting stuff. And the last future direction that I was interested in, I think it would be really interesting to further explore the possible protective benefits of using antipsychotic medication early on individuals who suffer from substance-induced psychosis. I think this can have like really important clinical implications, and I think that it's something that should really be explored in order to try to make an improvement later on. So that concludes my talk. Thank you so much for listening and have a great day.